Shuttle and expression vectors. Shuttle vectors are those vectors that can replicate in two or even more unrelated organisms. So because of this reason they are called as shuttle vectors, क्योंकि इनमें ये एबिलिटी होती है कि ये दो या दो से ज़्यादा होस्ट ऑर्गेनिज्म जो कि आपस में रिलेट नहीं करते, दैट आर टोटली अनरिलेटेड, उनमें भी ये रेप्लिकेट कर सकते हैं. मिसाल के तौर पे बहुत सारे शटल वेक्टर्स ऐसे हैं जो कि यूज़ किए जा सकते हैं फॉर एस्चेरिशिया कोलाई और बैसिलस सबटिलिस के लिए. इसी तरह से इकोलाई या यीस्ट के लिए, या इकोलाई या मेमेलियन सेल्स में ये रेप्लिकेट कर सकते हैं. So because they can replicate between different unrelated host organisms, so they are called as shuttle vectors. Shuttle vectors, for example, if we are using them for Escherichia coli or the yeast, then there are certain limitations. One limitation is that origin of replication of bacteria or the bacterial plasmid, it will not work when the plasmid it will be used to transform yeast. So this problem can be overcome by the use of another origin of replication from the yeast. So such shuttle vectors, they contain two origin of replication, one from the bacteria and other from the yeast. Another additional segment that is added within the shuttle vector, especially when we are going to use them with the yeast, is centromeres, because centromere it is required for the binding of we can say that mitotic apparatus during the process of cell division. So it is very important for the division of DNA. So centromere it is an additional portion that is required. Another problem for using shuttle vectors for yeast is that the selectable markers that are used for the selection for bacterial host, they will not work with the yeast. मिसाल के तौर पे जो अगर हम एंटीबायोटिक यूज़ कर रहे हैं एज अ सिलेक्टेबल मार्कर्स, तो हो सकता है कि ये एंटीबायोटिक्स तो सिलेक्टेबल मार्कर के तौर पे एक्ट करें बैक्टीरिया के लिए, लेकिन यीस्ट में ये डिफरेंटली बिहेव करेंगी. सो सच एंटीबायोटिक्स selectable markers, they will not work for yeast because antibiotic mechanism may be different between prokaryotic nature of microbes or the eukaryote. So slight differences are there, so that the antibiotics that are acting on one host, it may be ineffective for another host. So because of this reason, the selectable markers of antibiotics for bacteria, they will not work for the yeast. For this reason, another selectable markers for the yeast like the synthesis of uracil, pyrimidine, purines, or other amino acids, they have been used for the shuttle vector that are used between E. coli and the yeast. This diagram indicated the genetic map of shuttle vector that is used for yeast. As I told you that it contains two regions of replication, one for the bacteria and the other from the yeast. And then ampicillin resistance gene, it is also their centromere that play a role in the replication of the DNA, and polylinker cloning site where foreign DNA can be inserted, and then some selectable marker for the yeast. They can also be there, and then some termination point for the transcription. They have also been incorporated, and of course promoters for the transcription of different genes. And this arrow indicates the direction of transcription. On the other hand, expression vectors are those vectors where the expression of the cloned gene it can be controlled. Because when foreign DNA or the foreign gene it has been inserted, sometime it will not express or it will express poorly. So The use of expression vector will provide advantage to the experimenter to control the expression of the control cloned genes. तो इसमें जो एक्सप्रेशन कंट्रोल किया जाता है तो ऑफ कोर्स इसमें जो इसकी जीन की ट्रांसक्रिप्शन है उसको कंट्रोल किया जाता है और इसमें जो स्ट्रॉन्ग प्रोमोटर है इसकी बड़ी अहमियत है. मिसाल के तौर पे अगर हम एक ऐसी जीन यूज़ करें जिसका अपना ही प्रमोटर है या नेटिव प्रमोटर है तो सम टाइम होस्ट ऑर्गेनिज्म में वो भी एक्सप्रेस नहीं करता. तो इस पर्पस के लिए ट्रांसक्रिप्शन को ऑप्टिमाइज करने के लिए कुछ प्रमोटर सेलेक्ट किए जाते हैं लाइक लैक प्रमोटर, ट्रिप प्रमोटर and PL promoter from lambda phages. They have been used to optimize the expression of the foreign gene. Again, this diagram, it indicates the genetic map of an expression vector which is BSE420. Actually, this vector has been synthesized or constructed by a commercial company like In Vitro Gene, and it contains restriction sites for cloning foreign DNA, and then it also contains a very strong promoter like TRC that can control the expression of foreign DNA that is inserted between these cloning sites. And adjacent to this promoter there is a LAC operator, and then ST or Shine-Delgarno sequences that will help to bind the messenger RNA after transcription with the ribosome. So Shine-Delgarno sequence can also be synthesized, and then there are some terminators like T1, T2 that can terminate the expression of the foreign gene. And some selectable markers in the form of ampicillin resistance are also there. And there is a LAC-I gene that can encode LAC repressor. And of course, one of the important region, the origin of replication, that will facilitate the independent replication of the expression vector into a suitable host organism.